Chapter 12 Biology, Video 6 Theory, Respiration. So now in O levels you have learned in respiration what happens is chemicals are broken down to release chemical potential energy, and this process is what keeps us alive, the cells alive. So because everything in our body requires energy, so this energy requiring processes will happen only if energy is made available to them. So respiration is the process that releases energy. So this process does not actually take place at once. We learned earlier that there are a lot of small steps that is involved, and these small steps release chemical potential energy step by step. The reason is that if all of the energy is released at once, the molecules in the cells would not be able to harness that energy, and most of the energy would get wasted. So to make sure that the energy is used as efficiently as possible, it takes place in several steps. So this energy is made available by breaking down organic molecules, and that chemical potential energy is used to synthesize ATP. The main fuel for most cells is carbohydrate, usually glucose. Many cells can use only glucose as their respiratory substrate, but others break down fatty acids, glycerol, amino acids in respiration. So that means even though glucose is the most common form of respiratory fuel, that doesn't mean the other materials cannot be broken down. They can be broken down as well. Fatty acids, glycerol and amino acids can be broken down, but they are a little bit of a more complex process, but will finally the usual products will be formed. Glucose breakdown can be divided into 4 stages: glycolysis, the link reaction, Krebs cycle and oxidative phosphorylation. And we have actually learned some parts of this. We have discussed this a little in our earlier lessons, but now we are going to discuss this in detail. So the sequence of events in respiration is described in figure 12.7. There are anaerobic and aerobic respiration. You know what they mean. So now here we are talking about inside the aerobic pathways in mitochondria, link reaction, Krebs cycle, oxidative phosphorylation take place. Then glycolysis is a common aspect for all of it, right? So the combinations of these things also you should understand in detail. Write down a note. Respiration. This is the process by which organic molecules are broken down to release their chemical potential energy, which is used to synthesize ATP. Glucose breakdown, next point, glucose breakdown can be divided into 4 stages. Number 1, glycolysis. Number 2, the link reaction. Number 3, Krebs cycle. And number 4, oxidative phosphorylation. The link reaction, Krebs cycle. The first one is glycolysis, then link reaction, Krebs cycle, and oxidative phosphorylation. Then glycolysis. This is the splitting or lysis of glucose, splitting of glucose. It is by itself, it is a multi-step process, right? So the respiration is a multi-step process, but at the same time glycolysis, these individual reactions will also have different steps in it. Glycolysis is a multi-step process in which a glucose molecule with 6 carbon atoms is eventually split into 2 molecules of pyruvate, each with 3 carbon atoms. So 6 carbon glucose is broken down into 3 carbon pyruvate. 2 molecules of pyruvate is being produced. Energy from ATP is needed in the first steps. So to produce energy is required. So in these first steps, ATP is being used up. But energy, so in the end, the net release of energy is more than the net gain of energy. But energy is released in later steps when it can be used to make ATP. There is a net gain of 2 ATP molecules per molecule of glucose broken down. That is, when each glucose molecule is broken down, 2 more molecules of ATP would be produced than the number of molecules, than the number of ATP molecules that would be broken down. Glycolysis takes place in the cytoplasm of a cell. A simplified flow diagram of the pathway is shown in figure 12.8. Let's go to 12.8 and see how it happens. So this is the glycolytic pathway. In glycolysis, the whole thing, what happens is glucose hexose, that is 6 carbon, it takes in an ATP to produce fructose phosphate 6 carbon, and that breaks down using another ATP to produce fructose biphosphate 6 carbon. This fructose phosphate turning into fructose biphosphate is known as phosphorylation. Then fructose biphosphate split into 2 to produce 2 molecules of triose phosphate 3CP, 3 carbons, and releasing 2 ATP and producing intermediates and finally producing 2 molecules of pyruvate. Here there is another process that takes place. We will be coming into that. This whole thing is known as glycolysis. Altogether 4 ATP would be produced, while 2 ATPs would be used up. Right? So there is a net gain of 2 ATPs. In the first stage, phosphorylation, glucose is phosphorylated using ATP. As we saw on page 269, glucose is energy rich but does not react easily. I hope you can remember that. So it is reduced by using, the reactivity is increased by using enzymes. To tap the bond energy of glucose, energy must first be used to make the reaction easier. 2 ATP molecules are used for each molecule of glucose to make first glucose phosphate. 2 ATP molecules are used for each molecule of glucose to make first glucose phosphate. Then fructose phosphate, then fructose biphosphate, which breaks down to produce 2 molecules of triose phosphate. Hydrogen is then removed from triose phosphate and transferred to the carrier molecule NAD, nicotinamide adenine dinucleotide. So this is a carrier molecule. How do we do that? Hydrogen is removed. We will be learning about this NAD a little bit later in detail. The molecule of reduced NAD are produced for each molecule of glucose entering glycolysis. The hydrogen carried by reduced NAD can easily be transferred to other molecules and are used in oxidative phosphorylation to generate ATP. The end product of glycolysis, pyruvate, still contains great deal of chemical potential energy. When free oxygen is available, some of this energy can be released via the Krebs cycle and oxidative phosphorylation. However, the pyruvate first enters the link reaction, which takes place in the mitochondria. So, basically what you need to understand, first part is the glycolysis. In glycolysis, there is a gain or net gain of 2 ATPs, net gain of 2 ATPs, and 2 reduced NADs are produced. How are these NADs produced? 2 hydrogens are removed in the conversion of triose phosphate into 2 molecules of pyruvate via different intermediates. Please write down a note. In glycolysis, through a multi-step process, hexose glucose is split into two molecules of pyruvate with a net gain of 2 ATP molecules per molecule, with a net gain of 2 ATP molecules per molecule of glucose broken down. Leave a line and write. Glycolysis takes place in the cytoplasm of a cell, different from inside the mitochondria. So you have obviously learned that respiration takes place inside the mitochondria, but now we are learning that this is taking place inside the cytoplasm. So this is just one part of it. Right, now I want you to draw this figure 12.8, pause the video and draw 12.8 and then come back to write the note. I hope you have drawn the note, so write down. In the first stage, glucose is phosphorylated using ATP. Within brackets, right? ATP is needed to make the reaction easier as glucose is highly stable. Close brackets. After phosphorylation, two molecules of triose phosphate are produced. From which hydrogen is removed and given over to carrier molecule NAD. Carrier molecule NAD. The reduced next point, the reduced NAD molecules are used in oxidative phosphorylation to generate ATP. Next point, the final product pyruvate will go through further reactions to produce more energy. Right. Then we come to the link reaction. Pyruvate passes, now the pyruvate produced as a result of the earlier reaction, glycolysis, is now it would come to the link reaction. It passes by active transport from the cytoplasm. Why glycolysis takes place in the cytoplasm? From cytoplasm through the outer and inner membrane of a mitochondrion. So it has it's a nuclear membrane. Not nuclear membrane, my apologies, it's an envelope membrane. So there are two walls. So the inner and outer membrane, through them, it will enter the mitochondrial matrix. it will be decarboxylated, that is carbon dioxide is removed, decarboxylated, dehydrogenated, that is hydrogen is removed, and combined with coenzyme A, CoA, to give acetyl coenzyme A. This whole thing is known as the link reaction. So you have pyruvate coming in. It is decarboxylated, that means carbon dioxide is removed. Dehydrogenated, hydrogen is removed, and combined with coenzyme A to give coacetyl to give acetyl coenzyme A. Coenzyme A is a complex molecule composed of a nucleoside, adenine plus ribose, with a vitamin, pantothenic acid, and acts as a carrier of acetyl groups to the Krebs cycle. So you can see one stage is dependent on the earlier one. So the link reaction is dependent on the glycolysis which occurred outside in the outside of the mitochondria, in the cytoplasm. The hydrogen removed from pyruvate is transferred to NAD. Right, so there is NAD to accept that hydrogen. So you have reduced NAD, carbon dioxide, acetyl coenzyme A in the end. So pyruvate combines it, goes through removal of carbon dioxide. That is, removal of carbon dioxide is known as decarboxylation. So decarboxylated, removal of carbon dioxide, hydrogen is removed, dehydrogenated, and then you get you have NAD to accept that hydrogen, and carbon dioxide is there as it is. Pyruvate, so the equation is, the Krebs cycle equation is pyruvate plus coenzyme A plus NAD gives acetyl coenzyme A plus carbon dioxide plus reduced NAD. Fatty acids from fat metabolism may also be used to produce acetyl coenzyme A. Fatty acids are broken down in the mitochondrion in a cycle of reactions in which each turn of the cycle shortens the fatty acid chain by a 2-carbon acetyl unit. Each of these can react with coenzyme A to produce acetyl coenzyme A, which, like that produced from pyruvate, now enters the Krebs cycle. So in breaking down of fat, that is in fat metabolism, the same thing happens. But there is no glycolysis. Here what happens is fat is broken down, reduced, the chain, the fatty chain is reduced by 2-carbon acetyl units each. In the end, it will produce acetyl coenzyme A. And after that, it goes into Krebs cycle. Write down the link reaction, write the note. Pyruvate produced as a result of glycolysis in the cytoplasm passes into the mitochondrial matrix by active transport. So that means it also uses energy. Leave a line and write. In the matrix, it will be decarboxylated, within brackets, carbon dioxide is removed, comma, dehydrogenated, within brackets, hydrogen is removed, and combined with coenzyme A. Within brackets C, capital C, simple O, capital A, to give acetyl coenzyme A. The removed hydrogen reduces an NAD. Then write the equation: pyruvate plus coenzyme A plus NAD gives acetyl coenzyme A plus carbon dioxide plus reduced NAD. Please write that down. Then leave a line and write fatty acids in fat metabolism. Fatty acids in fat metabolism shortens the fatty acid chain by 2 carbon acetyl unit each cycle to produce acetyl coenzyme A similar to what is produced by pyruvate reaction. I hope you understood that. After this, we will move into Krebs cycle.